Welcome, everyone. My name is Geneviève de Blois. I'm very happy to be hosting today's seminar. Just before we start, I would like to remind everyone that we will be hosting our 8th Canadian Conference on Epigenetic and IEC annual meeting of 2022. Early registration will open on July 4th and early bird deadline will be on August 3rd. The meeting will be held from the 4th to 7th of October 2022 at Esterel Resort in Quebec. So it is my great pleasure today to be hosting this last seminar before our summer break. I want to remind you that our next seminar will be held in September, on September 21st at 9 a.m. Pacific time or 12 p.m. Eastern time. And we will be hosting Cynthia Hawking from Sick Kids in Toronto. So today, it's my great pleasure to host trainee Benjamin Lebeau and PI Michael Witcher from the Witcher Lab at McGill University in Montreal. I would like to remind everyone before we start that if you have any questions, to please type them in the Q&A rather than in the chat. And I will be able to relay the questions to the speaker after their presentation. So our first presenter today is Benjamin Lebeau. Benjamin obtained his bachelor degree from the Department of Anatomy and Cell Biology at McGill University. He now studies as a PhD student in Dr. Michael Witcher's lab at McGill University at the Lady Davis Institute. His research focuses on the oncogenic and epigenetic impact of CTCF mutations or copy number loss in breast cancer. His project currently investigates the change in chromatin conformation that drive breast cancer progression following CTCF loss of heterozygosity, and also involves the development of a new bioinformatic tool to study the modification in DNA motif recognition following CTCF mutations. Following his PhD, he plans to move to Singapore to pursue postdoctoral studies that bridge artificial intelligence and epigenetics. So the title of his presentation today is Single Base Pair Resolution Analysis of the Altered DNA Binding Ability of Zinc-Finger 1 Mutated CTCF in the context of breast epithelial cancer. So Benjamin, you can start your presentation.
Thank you for the introduction. So yeah, I'm Ben. I'm a PhD student in Mike's lab, and I'm gonna start now. So we often think of our DNA as a sequence of ACGT that codes for gene, but our DNA is a molecule, and therefore it takes up space in 3D. So it needs to be stored and organized to be transcribed properly. The first level of this organization is the nucleosome, the chromosome territory, where each of the chromosomes in our cells would take up specific space in the nucleus, in which the mostly active part of the chromatin will segregate from the mostly inactive part of the chromatin in what we called A and B compartments. These compartments will then be further subdivided in continuous domains of increased interaction that we call topologically associated domains, or TADs, in which we can find smaller TADs called subTADs. TADs and subTADs organization is critical to the regulation of gene expression, as they promote the interaction between promoter and cis-regulatory element within the TADs, while insulating from element outside of the TAD. Therefore, defining where the boundaries of these TADs are involve which genes and which element can be regulated together and which cannot intervene in the regulation of these genes. And defining these boundaries is one of the functions of CTCF. making CTCF a very important multifunctional epigenetic regulator that is involved in blocking or promoting enhancer promoter contact, defining the boundaries between closed and open chromatin states. It's also involved in double-stranded break DNA damage repair, or it can be involved in the recruitment of the transcription machinery. And with all these functions, it makes CTCF an essential gene, as the biallelic deletion of CTCF is lethal in most cells. However, the single allele deletion is viable, but it comes with its issues, as a single allele deletion of CTCF is found in more than half of all breast tumors, and CTCF zygous mice will develop an increased rate of cancer. But today, I'm not going to talk to you about CTCF loss of heterozygosity. I'm going to talk about another dysfunction of CTCF that is found in cancer, being CTCF mutations. So two of the three most common CTCF mutations, being R377 and S354, are found around Zinc-Finger 4 of CTCF that is known to be involved in the binding of CTCF to its core recognition motif on the DNA. However, the third most common mutation of CTCF in cancer, H284, is located on the first Zinc-Finger of CTCF that currently is not well known if Zinc-Finger 1 of CTCF is directly involved in CTCF binding to the DNA, and if any motif is recognized specifically by Zinc-Finger 1. Also, interestingly, Zinc-Finger 1 mutation of CTCF are exclusive to breast cancer, and in which CTCF mutations are the second most enriched mutation in metastatic versus local tumors. And CTCF H284 mutations are also enriched in tumors resisting hormone therapy. Therefore, we decided to study the impact of CTCF Zinc-Finger 1 mutation on its binding to DNA and on cancer progression. So the first thing we did is we surveyed TCGA dataset to see what was the most common clinical genotype associated to CTCF Zinc-Finger 1 mutation. And we found that 84% of tumors carrying CTCF Zinc-Finger 1 mutation also carry a non-functional allele of CTCF, making the genotype where one allele has the mutation and one allele is non-functional, the clinically most relevant one, and one of the model we decided to study. Another genotype we decided to study is one in which the mutation of CTCF is inserted in both allele, making a cell line that expresses solely the mutated form of CTCF, but at a level that is similar to what we see in the wild type, therefore alleviating any confounding effect of the low levels of CTCF found in this model. Also, the alleviation of confounding effect is why we use MCF Trainee cells that are normal mammary epithelial cells that do not carry all the strong oncogenic expression found in breast cancer cells. So we did a CRISPR-Cas9 addition of our MCF Trainee cells to get all the models we wanted to study. And we did a ChIP-seq for CTCF on these cells to see if the binding of CTCF to the DNA would be changed by the introduction of the first Zinc-Finger mutation. And indeed, we found that about 3,800 sites were gained upon the mutation of CTCF, while about 6,500 sites were lost. And the changes in CTCF binding was very similar between our two cell lines, beside about 1,000 sites that were uniquely lost in the knock-in knockout cells, possibly due to the lower levels of CTCF. The next step of our analysis was to do a classical motif analysis to see if the recognition motif of Zinc-Finger 1 mutated CTCF is changed compared to wild type CTCF. However, in both our cell lines, whether the sites were constant, gain, or loss, the top motif that was found was the core recognition motif of CTCF that was also found in proportion that were very similar in all groups. Therefore, this validates what was shown before, that CTCF Zinc-Finger 1 is not involved in the recognition of the core motif of CTCF. However, since the core motif of CTCF is bound by Zinc-Finger 7 to Zinc-Finger 3, Zinc-Finger 1 would bind somewhere downstream of the motif. And if the change in motif caused by the mutation of CTCF is subtle, it could be lost by classical motif analysis, as it would be drowned by the consistency and the strong presence of the core elements within the binding motif of CTCF. So we needed a new tool that works downstream of classical motif analysis to analyze modification of a specific motif. And this is why, in collaboration with Kai-Chang Zhao from Celia Greenwood Lab, we developed MoMotif, or modification of motif, to work downstream of classical motif analysis and to find any changes for a specific motif between two conditions. So here we use MoMotif to compare the CTCF motif in the sites that were commonly lost in the Zinc-Finger 1 mutated cells, the site that didn't change between the mutant and the wild type cells. We extended the sequence of CTCF 30 base pair before and after the middle of the sequence, shown as the purple dot line here, that is the G at position 31 on the motif shown here. We saw that upstream of the motif, we didn't see many changes in the frequency of the bases, as it was the case also for bases very downstream of the motif. But when we looked at the bases that are just downstream of the CTCF motif, we could see big enrichment in the lost sites compared to the content sites that were highly significant. And when we turned these frequencies in a motif, we now see an extension of the CTCF motif specific to the lost sites that we defined as the A at position 4D, the G at position 43, and the C at position 46. And these three bases were partially present in more than half of all the lost sites, while they were excluded from the gain sites as less than 10% of them had the bases. Then we use a computational tool to predict the recognition motif of all Zinc-Finger of CTCF that we align to the motif we found with MoMotif. And we see that indeed Zinc-Finger 7 to 3 binds very well to the core motif that was found in the constant sites and the gain sites, while Zinc-Finger 2 and 1 are not necessary for the binding at these sites. However, in the lost sites, the sequence recognized by Zinc-Finger 2 and 1 aligns perfectly with the extension of the CTCF motif we found with MoMotif. Therefore, this hints at a mechanism where Zinc-Finger 1 mutated CTCF cannot bind the extension of, well, the extended motif of CTCF, as it requires a functional Zinc-Finger 1 to bind, and therefore it gets redistributed at other sites where the binding of Zinc-Finger 1 is not required. So then we wanted to see if the changes in binding of CTCF would lead to some changes in gene expression that could be linked to what we saw in the clinic for Zinc-Finger 1 mutated breast tumors. So when we did a RNA-seq and we compared the changes in gene expression between the control cells and the Zinc-Finger 1 mutated cells, we saw that gene expression was very similar in both cells, in which genes that were upregulated were strongly enriched for genes involved in the metabolism of drugs, which could be related to the phenotype of the tumors that are CTCF Zinc-Finger 1 mutated and enriched in tumors that resist hormonal therapy. And also genes that were down-regulated were enriched for genes involved in the organization of the extracellular matrix, the downregulation of which could have explained the increased metastatic rate of the CTCF mutated tumors. And then to relate the changes in CTCF binding to the changes in gene expression, we put our RNA-seq and our ChIP-seq data in a topological context of the chromatin with the IC data set we had in the wild type cells. And we did see a link between the loss of CTCF and the changes in gene expression that was very similar to the mechanism we saw in our CTCF loss of heterozygosity data set. So I'm not going to spoil it here, and you're gonna have to stay tuned for the next talk to know what this mechanism here is. But for here, in summary, we saw that CTCF H284N is unable to bind an extended motif of CTCF that we identify with our new tool motif, and the loss of binding of CTCF is associated with altered gene expression of genes involving xenobiotic metabolism and ECM organization that are associated to the phenotypes that are observed in CTCF mutated breast tumors. So that is all for my talk. So are the questions now, or do we move on to the next talk first?
We thank you, Benjamin. We will take questions now if there are some. So I'm going to start with a question from the audience, from Victor Corses. As ZF1 has also been shown to bind RNA. Are you planning to test this in your cells?
Yeah, so we did try to test it with an E-clip to see if it would change the, well, what are the RNA that binds to it. However, I couldn't get the EV-click to work, but yes, this is an avenue that we really want to try. Because also a part that I didn't go in details in the extension of the motif, there is the A and the G, but there's also the C here that is downstream of it, and that we don't see a direct link with the Zinc-Finger of CTCF. So it's possible that the coordination of the binding to DNA that Zinc-Finger 1 is doing is not just directly by binding to DNA, but it could also bind to an RNA that helps to recruit CTCF to it, or an RNA that recruits a protein that helps CTCF to bind, or directly bind a protein that helps its recruitment. So yes, it's an avenue that we want to analyze, but we don't have the result for that yet.
Very good. Thank you. I'm going to follow up with a question of my own. So I was, you might have mentioned this, but I was wondering if the presence of the mutation changes the complex formation for CTCF. And does it, were you able to detect this binding of the mutant CTCF or accessibility of these differential sites in tumors with different drug response, for example, or metastatic samples?
Yeah, so the data from the patients, we don't have a ChIP-seq in them, we only have the RNA-seq. So we couldn't do that type of analysis, but yeah, it would be a good idea to see how CTCF complex with itself when Zinc-Finger 1 is mutated, but we did not investigate this. But it is a very good idea.
Okay, thank you very much. So if there's no more question, we'll go to the second talk for today, which is, thank you very much again, Benjamin, for this very good presentation. So the next speaker is Dr. Michael Witcher. So Dr. Witcher obtained, just one second. I need to go back. Okay. So Dr. Witcher is an associate professor at the Faculty of Medicine of McGill University. His lab is located in the Lady Davis Institute, and he currently acts as an associate director of the Lady Davis Institute. He received his bachelor and master degrees from the Memorial University of Newfoundland, and he went on to pursue a PhD in experimental medicine at McGill University. Following this PhD, he carried out a postdoctoral studies at the Salk Institute under the mentorship of Dr. Beverly Emerson. Currently, research in the Witcher lab is focused on identifying epigenetic processes that are commonly deregulated in breast and ovarian cancer. Using this information, they aim to identify druggable targets and pathways to treat poor prognosis cancers. So the title of Dr. Witcher's talk is Epigenetic Reprogramming Drives Cell Invasion Programs. Dr. Witcher, you can start your presentation.
Okay, thank you, Geneviève. I will say that similar to the last presentation, that Ben really compiled most of the data that I'll be presenting here today, and all of the data that I'll be showing is unpublished, but is currently under revision. Okay. Okay, the slides are not, sorry, the slides are not progressing. Okay, here we go. Can you, just to make sure, can you hear me okay?
Yes.
Okay,
yes.
great. I turned off the laser pointer, and so I can't really explain this, but the last 3 times I used the laser pointer during my presentation, my computer crashed, and when I turned on the laser pointer, then I couldn't progress my slides. So my computer doesn't.
We can still see your regular cursor, I think.
Okay, so that's
Yeah.
fantastic.
Yeah.
Yep.
Thanks.
Perfect. Okay, breast cancer metastasis step process that necessitates a great deal of cell plasticity. And so because of this required plasticity, it has long been surmised that dynamic epigenetic processes may play a role in driving tumor invasion into the surrounding stroma and subsequent distal metastases. And Dr. Robert Weinberg has a great review on this. And while there's considerable evidence that epigenetic reprogramming, including histone methylation and DNA methylation, may drive tumor progression, it is less clear that changes to 3-dimensional chromatin architecture is also playing a role in tumor progression. I would say that this is an area that's really understudied in my opinion. As Ben alluded to earlier, the genome is partitioned into sets of large self-associating domains called topologically associated domains, or TADs. These large chromatin loops represent contiguous regions of DNA up to a megabase size that frequently interact with itself, but does not interact with regions outside the TAD. TADs are generally anchored and maintained by a protein called CTCF in collaboration with cohesins. These anchor sites act as insulators, preventing DNA from within the TAD from making contacts with neighboring TADs. And I'll interject here and say that TADs have a complex relationship with gene expression. And while it is widely expected, I would say, that disrupting a TAD anchor would wildly deregulate gene expression profiles. In fact, this doesn't seem to be the case. And so within TADs are found subTADs. These are relatively small loops, often representing contacts between enhancers and promoters, and are also sometimes dependent on CTCF. And there are some new manuscripts that have come online in the last year or two that show that CTCF is playing maybe an unexpected role in stabilizing promoter enhancer contacts. And within subTADs, beyond a role for stabilizing enhancer promoter interactions, CTCF also plays a contrary role, acting as an insulator that prevents incorrect pairing of promoter and distal enhancers. So as you might imagine, CTCF plays a complex role, I would say, in regulating gene expression, or potentially a complex role in regulating gene expression, which we'll explore to some degree in the talk that I'm presenting today. So we first became interested in exploring a role for chromatin architecture in regulating cell invasion and metastases after the publication of a really interesting 2018 Cell paper, where it was shown that clonal populations within benign breast tumors carrying a deletion in the region of chromosome 16q22.1 were prone to undergo evolution to become invasive. This greatly piqued our interest because this region carries the CTCF gene. And so we hypothesized that the loss of CTCF may lead to reprogramming of higher order chromatin architecture, thereby driving or facilitating tumor progression. And while many studies have confirmed an important role for CTCF in organizing 3D chromatin landscape, it's often overlooked that there's really quite some evidence that supports a tumor suppressive role for CTCF, and Ben alluded to this as well earlier. I guess most strikingly, CTCF heterozygous mice develop lymphomas, but the impact on solid tumor initiation progression remains very ambiguous. However, further supporting a role for CTCF as a tumor suppressor in humans, CTCF undergoes copy number loss in a spectrum of tumors, including over 50% of all breast tumors, indicating that lowering CTCF levels may have pro-oncogenic consequences. Of course, with any deletion in cancer, it's not just a single gene being deleted. And so in general, when clinicians or researchers observe a deletion of 16q22.1, they refer to this as a deletion of CDH1 or ecadherin. But as I'll show later in the talk, we think CTCF is really the primary target for 16q22.1, and that the phenotypes associated with that are more profound than the impact of losing ecadherin or CDH1. So our first step towards investigating a role for CTCF copy number loss and potentiating tumor progression was to generate relevant model systems. And right now we've generated 4 relevant model systems, 3 of which will form the basis of the talk today. First, to model CTCF copy number loss in normal mammary epithelial cells, we used CRISPR-Cas9 to knock out a single allele CTCF. This resulted in a lowering of CTCF levels by approximately 60%, 50 to 60, comparable to what we observe in PDX models of triple negative breast cancer, which is our second model observed here. The red boxes represent PDX tumors that carry very low levels of CTCF. And in fact, one PDX model, number 6, we have yet failed to confirm that it's expressing any CTCF. It must to some degree, I assume, but it's incredibly low, which was quite surprising. Third, we employed shRNAs to knock down CTCF and MCF7 cells. And finally, and in my opinion, this is probably the most interesting model, even though it's at very early stages, but we have recently engineered an inducible CTCF knockout model that, in collaboration with Josie Orsini-Siegel, also at McGill University, we have crossed with a HER2 model of breast cancer to directly examine the influence of CTCF on tumor biology in vivo. This project is really at its early stages and was delayed tremendously because of COVID. But I will say that based on our first litter of mice, which came just recently, we show that one allele loss of CTCF promotes greater tumor initiation and outgrowth in about 50% of the HER2 expressing tumors. And we have not had the opportunity to examine metastases in these models yet, since the data is so recent. So those models and that data is forthcoming. So first, we found that restoring physiological levels of CTCF through ectopic expression in conditionally reprogrammed triple negative PDX-derived lines really restricts the invasion of these triple negative breast cancer lines, supporting a role for CTCF in promoting cell invasion. Likewise, when we knock down CTCF in MCF7 cells, this strongly potentiated cell invasion through matrigel. So the model that will serve as the bulk of the talk that I'm going to give today is our MCF 10A cells that carry a deletion or a knockdown of CTCF through deletion of one allele, which we engineered using CRISPR-Cas9. And we found that these cells that carry only one copy of CTCF acquired a very striking capacity to invade through matrigel. Add back of CTCF compromised this capacity, indicating that the process is reversible. We also observed that these MCF10A cells gained the ability to form large, highly filled mammospheres that also are indicative of tumorigenic potential. That being said, when we first created these cell lines, they held potential to form tumors in immunocompromised mice. But we were a little bit frustrated that over time they appear to have lost this ability, or at least the penetrance has diminished, and underscores the necessity of incorporating transgenic models into our studies moving forward. As I mentioned previously, another tumor suppressor that resides on 16q22.1 is the classical tumor suppressor CDH1 or E-cadherin. As a comparison, when we knocked out either one or both alleles of this gene in MCF10A cells, we saw no impact on cell invasion or any other oncogenic program effect. And this further supports the concept that CTCF may be the primary target, or at least a very important target, for 16q22.1 deletion in progressing tumor biology. To define the transcriptional programs that might be driving the invasion of these CTCF depleted cells, we carried out RNA-seq analysis, which revealed an enrichment for the upregulation of genes involved in PI3 kinase signaling and cell adhesion programs. These genes, at least the ones that we think might be very important, include the cell surface receptors, FGFR1, HERB3, which encodes HER3, and the transcription factor SNAIL, commonly known SNAI1, commonly known as SNAIL. And I'll highlight SNAIL because it does prove to be a very important mediator of invasion in the system. We are also pleased to find that breast tumors carrying CTCF copy number loss are enriched for alterations to genes related to PI3 kinase signaling based on TCGA data, adding clinical relevance to our in vitro findings. To begin to gain insight into the mechanism whereby CTCF might control oncogenic pathways involved in cell invasion, and in particular the PI3 kinase pathways, we carried out ChIP-seq for CTCF. And we found that a total of approximately 5,100 CTCF sites were lost in our MCF10A single allele knockout cells, and that these sites were enriched in regions surrounding PI3 kinase genes, indicating that in normal cells, CTCF plays a previously unknown role, but important role in limiting the expression of genes involved in this proto-oncogenic signaling pathway. We subsequently moved on to validate whether indeed the PI3 kinase pathway was aberrantly activated in MCF10A CTCF heterozygous cells, and is shown by the Western blot here in the left for phospho-4EBP1 and phospho-S6K1, important downstream mediators of and transducers of PI3 kinase signaling. We see that both these proteins are indeed highly phosphorylated, indicating that the PI3 kinase pathway is indeed active to a substantial degree in these cells. On the right, we show immunofluorescent staining that shows increased activation of the PI3 kinase in the highly proliferative outer zone of these large mammospheres, as indicated by staining for phospho-S6, a target of S6 kinase. Again, in all situations where we examine this, we see that CTCF loss does potentiate PI3 kinase signaling. So this led us to test whether MCF10A cells carrying a single allele loss of CTCF might be sensitive to inhibitors of PI3 kinase signaling. To test this, we employed TORN1, an inhibitor of the mTOR complex, which is, again, another important downstream effector of PI3 kinase signaling. And we see that CTCF heterozygous cells are acutely sensitive to mTOR inhibition in the low nanomolar range, and these concentrations of TORN1 acutely impair the ability of these cells to invade through matrigel. And we see that this loss of invasiveness is tightly linked in panel D here on the right to translational repression of snail after exposure to TORN1. And we think snail in particular is an important oncogenic target that's upregulated after CTCF loss. Based on TCGA data, we see a clinical correlation between low CTCF expression and high snail expression. And we now know that this classical oncogene and important translational target of the PI3 kinase pathway is indeed an important mediator of invasion in the system, as snail knockdown, shown here on the right, dramatically reduces invasion of these CTCF heterozygous cells. Whoop, sorry, I skipped one there. So we next asked whether loss of CTCF leads to reprogramming of epigenetic marks at these altered genes. So we initially carried out a survey of epigenetic marks using CHIP qPCR at genes both upregulated and downregulated in CTCF plus minus cells. And we found that changes to mRNA levels correlated with altered activating marks, but with few concomitant changes in repressive marks. So thus we proceeded to carry out ChIP-seq for the activating marks H3K4 trimethylation and H3K27 acetylation using control MCF10A and CTCF heterozygous MCF10A cells. What we observed was a correlation between changes to both H3K4 trimethylation and H3K27 acetylation at genes undergoing transcriptional changes. This includes a loss of these activating marks at downregulated genes and a gain at upregulated genes. However, amongst these two marks, we found that H3K27 acetylation proved to hold the most statistically significant correlation with changes to gene expression. And we think that this is quite important because, as many of you are aware, H3K27 acetylation is considered to be a mark of active enhancer regions and is enriched at inactive and promoters, which are generally in contact with active enhancers. And we think that this is really important for the phenotypes that we are observing in these cells, and we'll go into some more detail about that in a few slides. In stark contrast, on the right, data shown for our 850K EPIC array to probe for correlations between gene expression alterations and changes to DNA methylation status. And surprisingly, we saw no relationship between gene expression changes and altered DNA methylation profiles whatsoever. This was a little surprising because historically, there's really been quite some data out there in the literature indicating that loss of CTCF binding may disrupt normal DNA methylation profiles, and that CTCF may be an important regulator of DNA methylation programming, but we certainly didn't see that in MCF10A cells. Okay, in the next slide here, I show a visual snapshot of our ChIP-seq data at the SNAIL1 gene. SNAIL1 nicely represents our model where loss of CTCF here just upstream of the SNAIL1 promoter with the dark purple line indicating MCF10A heterozygous cells. And we see a specific loss of CTCF at the site. And it leads to an activation of SNAIL through an accumulation of activating histone modifications throughout the promoter region. And even beyond that, at an active enhancer, which is potentiated by this loss of CTCF, which I will show again in a few slides. So next we asked whether these epigenetic reprogramming events and activation of pro-invasive transcriptional programs might be associated with changes to the three-dimensional chromatin landscape. It is challenging, at least it's challenging for us to predict how a partial loss of CTCF might impact chromatin architecture. We might expect to see a loss of TADs and subTADs. However, there are other proteins beyond CTCF that are important for maintaining TAD and subTAD structures. But the loss of CTCF sites harboring insulator activity might also be expected to allow a gain in promoter enhancer context leading to de novo subtads. So going into these experiments, we really didn't know what to expect. And so we went in with an open mind. To map long-range chromatin contacts across the genome, we use the Hi-C approach. As I'm sure many of you know, Hi-C works through sequencing DNA contacts formed across the genome through ligation of proximal fragments generated by DNA digestion. The resultant DNA contact matrices may be mapped through bioinformatics, and we use, for most of our research, a program called JuiceBox. We did compare this using independent bioinformatic softwares, and we found very consistent data. And this may be visualized using heat maps, seen here on the left, or pilot plots, and I'll show a number of pilot plots in the forthcoming slides. So on this slide, I demonstrate our Hi-C data spanning 6 megabases may be used to reveal topologically associated domains, basically large DNA loops that are flanked or anchored in reality by CTCF sites, shown here as blue arrows, that prohibit interactions in cis with regions outside the TAD. As you can see, the TADs appear visually as large red triangles bordered by a solid black line. And these TADs are not interacting by definition with neighboring TADs. So in the next slide, I show an example of how our Hi-C data, again acquired using the bioinformatics tool JuiceBox, may be used to identify subtads. It's seen here on the left as blue triangles. The DNA may interact in cis within these triangles or within these small loops, these subtads, but not with neighboring subtads. And this can be seen very clearly on the subtad on the right, where there, or on the TAD on the right here, demarcated by a dashed black line. And within this TAD, there are at least 2 subtads. And we can see that these are very distinct from one another, and they self-interact, but not with each other. And one of the limitations of Hi-C is really the resolution that we can really zoom into, and it's challenging to call subtads. But with new techniques and new bioinformatic tools, one of which I'm going to show again later, we are overcoming these challenges. Subtads in particular are known to be more dynamic in nature than TADs, and TADs are known to be, or generally thought to be, highly conserved across tissues. So we do hypothesize, or we did hypothesize going into these experiments, that changes to subtad organization may be the primary facilitator of gene expression changes in our CTCF knockout cells. So we observed that the genome of CTCF hemizygous cells, and this was a little bit surprising, I must say, because of the known role for CTCF and DNA damage repair, but CTCF hemizygous cells were quite stable on the chromosomal level and on the level of DNA compartments. We found only one gained interchromosomal interaction, seen here as a red box between chromosome 1 and 4, but this rearrangement was not found to coincide with a region where changes to gene transcription were seen. On the right, we see Hi-C data of the entirety of chromosome 20 with very consistent large TAD organizations between control and knockout cells. So next, we zoomed in to quantify potential remodeling of 3D chromatin on the level of TADs and subtads. And we saw reorganization at both the TAD and the subtad level, with 17% of TADs being reorganized and 28% of all subtads being rearranged. Next, we want to know whether the the disruption of TAD or subtads led to changes in chromatin insulation, or in other words, whether a loss of CTCF binding leads to aberrant DNA contacts. And we quantified the insulation at CTCF binding sites, and this can be visualized using pilot plots seen below. I apologize for the quirkiness of my cursor, but it's appearing and disappearing on my screen, so I lose track where it is on occasion. Sorry. So yeah. So sorry. The pilot plots shown here represent a composite of all the sequencing reads across the genome at specific types of loci. Here, we're looking at DNA contacts at CTCF sites, either at constant or lost sites. The two red triangles on the right and the left of CTCF site number 1, CTCF sites are demarcated with a green arrow, indicate DNA contacts surrounding the CTCF boundary. So as you can see that at CTCF sites that remain constant in our MCF10A cells, that there are very few gain or loss of interactions crossing these boundaries. The hemizygous cells can be seen here as arrow number 2. However, next, if we look at CTCF's lost sites, shown as arrow number 3 in MCF10A cells, we see that these CTCF sites are weaker insulators under basal conditions, as indicated by the lighter blue color between the adjacent TADs and subTADs. In MCF10A heterozygous cells at CTCF sites that are lost, shown by arrow 4, we see a gain in de novo DNA contacts spanning the 5' and 3' regions of the previous CTCF binding sites, indicated by these red and pink dots above the arrow. So next, we subdivided all of these self-interacting regions that flanked CTCF sites that were lost in MCF10A cells into categories of TADs or subTADs. At TADs, we see that a disruption of CTCF binding, shown by arrow number 6, results in only a small gain of interaction across boundaries, indicating that TAD boundaries are still largely intact regardless of CTCF binding. However, in contrast, at lost CTCF sites adjacent to subTADs, shown on the right by arrow number 8, we see a tremendous gain of DNA interactions across this former boundary, as indicated in an increase in red dots above arrow number 8, seen here. And we also see that the insulation score drops dramatically for these subTADs. And so again, if you compare arrow number 7, which represents CTCF in wild type cells at subTADs, we see that there's a leakiness at subTADs. And so the insulator activity at these subTAD boundaries is generally quite weak. And when these sites are lost, we see a large number of gain de novo contacts. So importantly, when we map these rearrangements to genes that were deregulated based on our RNA-seq data, we see that de novo gains in subTAD contacts were especially correlative with genes and with changes in gene expression, especially with genes that were upregulated. Not surprisingly, pathway analysis revealed of these upregulated genes revealed that de novo subTAD contacts were enriched surrounding genes of the PI3 kinase pathway. And so we think that a gain in subTAD contacts are really driving gene expression changes, and especially upregulation of genes in cells carrying single allele CTCF loss. And through mapping these de novo contacts, we also know that they commonly represent de novo promoter enhancer interactions. And again, back to my favorite new gene, snail, an excellent example of how de novo enhancer promoter contact driving the expression of an oncogene in CTCF heterozygous cells may be found at snail locus. Panel A shows our Hi-C data across the locus, with the blue panel on the far right zoomed in to encompass a 200,000 base pair region surrounding the snail gene. And so this analysis in particular was carried out using a pipeline called Hi-Fi, which was developed by the lab of Matthew Blanchette at McGill University, and allows a more refined resolution of Hi-C than typical pipelines such as JuiceBox. Using Hi-Fi, we detected a single de novo contact at the snail locus in our CTCF heterozygous cells, which represents an interaction between snail and a downstream enhancer. And so we confirmed that this enhancer was active as indicated by an enrichment for the histone modification H3K27 acetylation. Panel B shows our ChIP-seq data, which indicates that the downstream enhancer is silent under basal conditions, but becomes active in CTCF hemizygous cells, and likely is characterized by a new DNA contact. On the left in panel C, we validated that snail expression is directly regulated by CTCF through the use of a catalytically inactive Cas9 construct, where we use that construct to disrupt CTCF binding just upstream of the snail promoter, and we found that this leads to a robust upregulation of snail transcription. And so thus, overall, our models that in normal tissue, weak CTCF sites within TADs act as insulators, preventing enhancers from aberrantly activating genes involved in cell migration and evasion. Upon CTCF copy number loss, these weak sites are compromised, allowing de novo enhancer promoter contacts cross these insulator elements and drive the activation of formerly silenced oncogenes. So in sum, finally, I would like to thank members of my lab, especially Benjamin Lebeau, really played a major role in driving this project, and he'll be greatly missed when he moves to Singapore to pursue a postdoc. And of course, I'd also like to thank the many funding agencies that generously contributed to this project. And thank you for your attention.
Thank you very much, Dr. Witcher, for this amazing presentation. It was very great data and very interesting talk. I'll start with the question from the audience, from Jing Han. Thanks for your wonderful talk. Did the low correlation between DNA methylation and TAD is specific for cancer type, or do you expect this to be a general observation for all tumor types or cancer types?
Yeah, so that's a good question. And so right now, especially using our transgenic models, we're hoping to be able to answer questions about tissue specificity of the control of CTCF on subtads and on epigenetic changes. And so I don't think our data in any way discredit previous studies where they found that CTCF controls DNA methylation. But I think in this context, in normal tissue, it very well may be a tissue-specific effect. And certainly in any of the breast epithelial lines where we've looked, we did not see a profound effect of CTCF on controlling DNA methylation. And so we think that there are independent forces at play.
Okay, thank you. We have another question from Suji Yang. I feel so surprised that CTCF shows the highest mutation in uterine cancer. Do you know any study focus this specific disease?
Yeah, so the only one that I know, yeah, so same with, so I'll say two things. When looking at the mutation rate of a protein in a particular type of cancer, it's a good idea to look at the mutation of other proteins in that particular cancer. And so uterine cancer as a I would say a monumental mutational load. And I think this is because of defects in mismatch repair genes. And so many genes are mutated in uterine cancer, not just CTCF. So I would say with that complex mutational background, it's challenging to figure out in uterine cancer exactly what CTCF mutations are achieving. And that's one of the reasons that we really like MCF10A as a model. There are really very few defects in these cells. There's the deletion of CDKN2A or the INK4 locus. But apart from that, this is genomically stable cell line. So it's easy to tease out the effects of mutations or different perturbations. There is an oncogene paper published by a group from Australia where they do look at the impact of CTCF mutations that are found in uterine cancer. They do this simply by ectopic expression, and they find that it may have some impact on survival and proliferation. So obviously that's not the ideal way to study these mutations. We're pretty excited about knocking these mutations into lines using CRISPR-Cas9. But again, they did have an impact just by ectopic expression. So it may be on impacting survival or mutation. We think most of these mutations are impacting progression and survival. And especially if you look at Zinc-Finger 1, if you look at where it's enriched, it's really enriched in metastatic disease and in drug-resistant disease. And so we think that this is probably more important for progression rather than initiation.
Very good, thank you. I'm going to ask one of my own questions as well. So I was wondering, given the effect that you see on the PI3 kinase gene or PI3 kinase signaling increase that you observe, that may also relate to mTOR activity as you're treating also with taurine. I was wondering if you have any idea of whether these cancer cells or breast cancer cells actually display a different metabolic profile, given the role of these signaling in regulating metabolism, and how this could be linked to maybe drug response or drug resistance.
Yeah, so we published a paper in 2015 that links CTCF to altered metabolomic profiles and as being a regulator of metabolism. I think for sure, yes, that there is an altered metabolism in these cells. We haven't, let's say, until we generate or we have our mouse models up and running, I'm going to avoid that just for the sake of complexity. And so, related to the work you're doing, Genevieve, what I'm really interested in understanding is, maybe that this predicts some therapeutic vulnerabilities, even though, so we did carry out a CRISPR-Cas9 screen on these MCF10A cells, and we did not find many metabolomic regulators. So that sort of tempered our enthusiasm about that. Mm-hmm. But I am quite interested in knowing whether or not, these changes in epigenetic marks that we find are maybe coupled with changes in metabolic programming. And so we think they're coordinated for sure. Because loss of CTCF is clear, and we didn't go into this, but we know that it's controlling translation, right? So there's a link between loss of CTCF and translational control through the PI3 kinase pathway. We've done ribosome profiling, and so polysome profiling. So we know there's a link there. So we think that there's a link there with energetics as well, but and that may play a role in the epigenetic reprogramming that we're seeing. So we're quite interested in that, but I want to wait to do that in our mouse models because I think it's far more powerful.
Yeah, that makes sense. Thank you. One last question. Is the mutation more frequent in a specific subtype of breast cancer?
Yeah.
Or the loss or increased expression?
I can't. So yes. So CTCF mutation, and one of the reasons that we're not actively pursuing CTCF mutations to let's say a greater degree than we have. We use these models really as a tool to allow Ben and Katie to develop motif, which is I'm really quite excited about, because Ben didn't highlight all of its capabilities, but you can also take a ChIP-seq set and subdivide those data into like CTCF or any transcription factor binding site at promoters or enhancers or proximal to subtads or proximal to tad boundaries, and look and probe for motif differences. So we think the tool is quite important. CTCF mutations are found primarily in luminal breast cancers. And so, luminal breast cancers they're not cured, but we have very good treatments for them. So we don't want to really go down that route and try to understand the role of CTCF mutation in driving luminal breast cancer because it's practically a cured disease. What is interesting is that CTCF Zinc-Finger 1 mutation is enriched in hormone-resistant breast tumors. And so, based on Ben's data, we saw that those cells carrying CTCF mutation activate a drug metabolism program. And so we think, well, that's probably the reason that CTCF Zinc-Finger 1 mutation is enriched in hormone-resistant breast tumors, because it potentiates the activation of this drug metabolism program. And that may be relevant to other cancers as well. We don't know.
Okay, thank you very much. So we'll end the meeting here. I want to thank both speakers again for these very amazing presentations. And I want to remind everyone that we will take a break over the summer, and the CERC seminars will be back on September 21st. Thank you very much.
Genevieve, do you want to mention the larger meeting again for
Oh,
anybody?
yes, I can share my screen again. So just to remind everyone, we're going to have the 8th Canadian Conference on Epigenetics and Hayek Annual Conference that will be held on October 4th to 7th, 2022 at Estella Resort. Registrations will open on July 4th, which is really soon, and early bird deadline registration is August 3rd. Thank you.
Thanks, everyone. Bye.
Thanks.
Have a great week.